Hello, honors bio students. This is the third time I've tried this video, so here's hoping it works this time. Been having some technical difficulties. I am hoping to continue the conversation we had in class. I wanted to give you a quick review for osmosis, which can be a slightly frustrating topic, and it's understandable. So I drew a little picture over here of an animal cell, and just to give you kind of a starting point, the inside of the cell is 99% water and 1% glucose. The outside of the cell is 96% water, 4% glucose. Now, what you want to be thinking about, and if you need to, hit pause so you can look at your notes. You want to be thinking about the outside of the cell. Is that outside, is that blue, what I highlighted, is that a higher concentration of solutes, the glucose, or is that a lower concentration compared to the inside? So I'm going to ask you a couple of questions here. I want to know what kind of solution this is. Is this a hypertonic, a hypotonic, or an isotonic? And then I want to know what direction the water is going to move. Is the water going to come in, or is the water going to leave the cell? So as you hopefully paused and looked at your notes, and as you reviewed, you should have found that this is a hypertonic solution. There is a greater amount of solutes in that solution, and the water should move out of the cell. This is the high concentration of water. This is a low concentration of water. Remember, glucose would move inside, but it can't. It's nice and big and polar. So hopefully that gives you a chance to review that a little bit. Now, just to continue the video that I made over the weekend, facilitated diffusion, you remember that it is high to low concentration. It's still passive. It doesn't need energy to happen. And the one thing that we added was a protein that could span the membrane and create a tunnel that allowed molecules to go through. Now, the one thing I did forget to add is that this is very specific. So I made a quick little membrane here, and each one of these red transport proteins, it's going to allow a different kind of molecule through. The one on the left would allow this green circle. Not very complicated molecules there. The one in the middle would allow for these little green triangle molecules. And in this case, the high concentration is on the inside, and it would go outside, just to give you something different. And then the third example, let me use purple, would be using our little blue squares. And then on this one, the high concentration would be in the outside, and it would be moving in. And oftentimes the way these work is that the shape of the transport protein only allows that kind of molecule through. Sometimes it also has to do with the charge and the interaction of the amino acids and the protein, but it's specific. That's one thing that this diagram didn't kind of give you an idea of, but they are specific. Now, active transport is going to be an entirely different ball game. In active transport, we want to concentrate molecules. In this case, we're going low to high concentrations. Now, this does not happen naturally, so this is an active process. You need to add energy to this system. Now, the energy that's going to be used is that ATP that you heard about previously, that adenosine triphosphate. That's going to give it a little bundle of energy in order to change shape and move a molecule across the membrane. So you're going to add energy. These are also highly specific, and so they're going to use a protein. Oftentimes these are transport proteins, but they oftentimes are called pumps. For instance, the classic example here would be the sodium potassium pump. And that's kind of how you say it, the sodium potassium pump. And annoyingly, the Na is sodium These are the Latin and Greek derivations, sodium, and the K is potassium. And so happen to be the two elements that don't have the first letter as their symbol. So the sodium potassium pump, and what it does is it pumps 3 sodium outside the cell and 2 potassium inside the cell. And it just keeps doing it. That's its job. It just repeats. And so notice in this diagram, it just circles around. It just keeps doing this. So you want to treat this as like a step by step by step. So if my little yellow guys are my sodium and my little green guys are my potassium, the little squares. So think about what's going to happen. They're going to pump out, oops, sorry, change colors there. They're going to pump out the sodium, and if they keep doing this, if they keep pumping more sodium, we're going to get a high concentration of sodium. And if they keep pumping the potassium in, we're going to get our high concentration of potassium on the inside. And because this repeats, it keeps going and going and going, you end up with a high concentration of sodium on the outside, a high concentration of potassium on the inside. Now, this is extremely important in the neurons of our cells and animal cells. And I'll give you kind of a weirdly fascinating idea here. If you've ever watched murder mysteries or forensic science shows, one of the things that they sometimes will say is, oh my gosh, he's been poisoned. Well, one of the poisons out there is a poison that basically sits right in this little pocket here, or it might sit on the outside. And as it does that, it blocks the transporter. And if you block it, then in essence, you've locked that door and these guys can't get in and can't get out. And if you lose that ability, then your neurons basically are done. You can't send signals and you die. Your heart stops, your brain stops. It's a pretty gruesome affair. But, and some animals, their venom works like that as well. Another example here is a proton pump, and these are pretty cool. We see these in plant cells. We also see them in lysosomes where we want an acidic solution. And so what we're doing, we've got a hydrogen ion in a low concentration. The little explosion here is ATP, same thing up here, ATP. And it is going to give energy for this proton pump to move it across the membrane. And these protons, they're hydrogen ions, and this makes it acidic. Now, usually it's not going to be like pH 1, although the stomach is. But a lot of times in cells, it just might make it like pH 4 or 5, like lemon juice. And so these proton pumps, they're actively transporting it, and you're kind of creating a acidic situation. So if we had a lysosome here, and we saw this in amoeba that was eating something, these proton pumps are going to make this more acidic on the inside. That's going to make those enzymes work better in this case. So that gives you a nice a second example. Now, I want to end with just a quick review to help you kind of distinguish between these. See if you can match. So we've got A, the purple, B with the triangles in blue, the triangles in orange and the blue circles. Either one of those is the same. And then C, which is our third one. And so you can hit pause if you need to. I don't want to put you on the spot. You can hit pause and make sure that you understand. You want to think about where is the high concentration? Where is the low concentration? Is there energy involved? Look at the arrows and see where they're moving and see if you can match up. Which one would be showing diffusion through the membrane? Which would be showing active transport with energy, and which one would be showing facilitated diffusion. Well, hope this helps, and that'll let us hit the ground running and get to the lab that we want to get to as well. So don't forget to study for your cell quiz, just the pictures of the animal cell and the plant cell. So have a lovely night.